Hello students, this is Dr. Faizan Mirza. I'm discussing with you the solved MCQ paper of Feb March 2022. This was the first paper in which 1 hour 15 minutes were given, and from now on the MCQ paper of 9700 will be of 1 hour 15 minutes. There will be 40 questions. Each question will have four options A, B, C, and D, and you'll have to select one option for each question, the correct option. The paper format is pretty much the same, but yes, the difficulty level is slightly raised. There will be more questions regarding logical reasoning and new cases. In most cases, there are questions where are three closed options and you have to select the one which is correct. Unlike previously where we used to have two options which were outright wrong and you had to choose between two. So that's the kind of difficulty that they have raised. But since you have another 15 minutes now, so that's not going to be a big problem for you. Make sure that you keep on annotating the question, whichever you are discussing or whichever you are solving. So let's start with the working. I'll be discussing question 1 till question 20 in this video, and the remaining half of the paper will be discussed in the next video. So guys, this is Feb March 2022. I'm discussing the details of these questions. As I've already mentioned that the difficulty level of this paper is a bit raised than as compared to the previous papers. Even if you see the specimen paper, so this is a bit different from that, because in specimen paper they gave some very easy questions and they were not at all of the same difficulty level which we actually expect now for a 1 hour 50-minute paper. So starting off with the question, again, the format is same, pretty much the same, that the first 20 questions are from the first 6 chapters and the remaining half of the paper will deal with question from the latter half of your syllabus. So question 1 is about cell and microscopy. You are given magnification of 400x and then you are asked which of these will be clearly visible. We note that the ribosomes are just not visible at all. I have annotated. I recommend all my students to keep on make sure that you also annotate whatever you know about those facts. So just recall and state so that it's easier for you to pick on the answer. So in this case, this which structure will be visible or not. So ribosomes, we know it's not visible in light microscope at all. Endoplasmic reticulum is again not visible at all in the light microscope. Centrioles are not visible in the light microscope either. Chloroplasts are the only options which are visible at a high power, and 400 magnification, this just be visible. So that will be the answer for our question 1. Question 2 is about microvilli and Reuter cell, which characteristics they are saying are identified by these statements. We know that the Reuter cell is having a cell wall around it. Yes, there is presence. Reuter cell can have a cell wall surrounding it. Microvilli with cell wall is actually not an option. Microvilli can never have cell wall around it because microvilli is a feature of an animal cell and not a plant cell. So microvilli just can never be in the cell wall. So you have to choose from either A or from C. Then coming to more than one present on the cell, we know that they are asking about microvillas. Microvillas means this is plural and this is more than one on cell. So we know microvilli are more than one. So so you can just see this that yes, present more than one. They are asking about plural. Microvilli are plural and more than one. So yes, that's the answer. Root hair cell is a singular term. So we can't choose this for more than one because each cell is having one root hair cell, each plant cell. So our answer will be A. The photomicrograph of plants has 25 micrometer in width from X to Y. So this X to Y is 25 micrometer. If this X to Y is 25 micrometer, this means this is actual width given to you. You apply the formula magnification of the image upon actual. Measure the length of X to Y in centimeters from here to here. Just place your ruler, measure it in centimeter, multiplied by 10,000. The answer comes in micrometer. Once you have the value, this will be your image. You will keep this value as a numerator and divide it by actual. Your actual is 25 micrometer. You just divide these and you get the answer. Make sure that you just see which exponent you have to work on and you can answer that accordingly. Question 4, in which eukaryotic cell structures contain nucleic acids? We know nucleic acids could be DNA or RNA. So which cytoplasm can have these nucleic acids? Golgi body will not have. Mitochondrial nuclei can have. So Golgi is not having. So an option with Golgi is wrong. Similarly, centrioles, there is a wrong option in B. In C, again, centrioles are wrong option. Remaining options are correct. So, but if you come to option D, chloroplast, mitochondria, cytoplasm, ribosome, they all have DNA and or RNA, ribosomal RNA is present. Sometimes I think some of the students might be confused at how a ribosome can have nucleic acid. So it can have it actually is having ribosomal RNA, or it can have mRNA during transition, and it can have tRNA as well. But yeah, basically it's ribosomal RNA that you need to make sure that you follow that. Okay, this is the nucleic acid present in the ribosome. So question 5. Which statement about viruses are correct? So which statement is correct? They contain DNA or RNA. Yes, if this was mentioned and, then this would have been a wrong statement, but or is the correct option. They use host cells as virus proteins. Correct. They have a protective coat of peptidoglycan. No, they have a protein coat. Peptidoglycan is a feature of bacteria. So 1 and 2 are correct. We select the option B. Question 6. What are the features of prokaryotic cell, chloroplast, and mitochondria? You have to select the common feature in them. We know circular DNA is present there. Ribosomes are smaller than 80s, and these are 70s ribosomes. They are present, surrounded by a double membrane. Okay, so chloroplast and mitochondria are surrounded by a double membrane, but a prokaryotic cell is not surrounded by a double membrane. So D cannot be the answer. Our answer will be B. A student used a Benedict solution to test a sample of known carbohydrate. Known carbohydrate. Okay. At the end of the test, solution was blue. Means me reduced sugar was absent. Which carbohydrate could be present in the sample? So all our options here: fructose, glucose, maltose, they all are reducing sugar, so they all must be present. The only option where you're not reducing sugar here in all these is D. So that's actually the answer. Sugars with a ring structure can also have a linear structure. This is a linear structure shown to you. You can just number the carbons and see how many carbons it's having. So it's 1, 2, 3, 4, 5, and 6 carbons are here. So which sugar molecules could be represented by the linear structure in the diagram? Could it be a ribose? Could it be deoxyribose? Could it be glucose? We know ribose and deoxyribose, they are rings. We know that glucose is linear and ring both, because glucose can exist in both conditions as linear as well as ring, whereas ribose, deoxyribose, they are pentose sugars, 5 carbon in DNA and RNA. They exist only in rings. They do not exist as linear structures. So answer will be 3. Which row matches each molecule to the type of bond present? So ester bond, we just recall ester bond is present in lipid and fat. We know it's present in lipid and fat. The hydrogen bond, they are present in protein and cellulose. You can just recall this. Disulfide bonds are present in protein. So just look for this. Amylase is a protein, so it cannot have ester bonds. So that's the wrong option. Again, glycerol is just an alcohol. It cannot have an ester bond until it's bonded to a saturated or an unsaturated fatty acid. Similarly, glycogen cannot have hydrogen bonds because it's neither protein nor cellulose. Come to lipids. Yes, lipids can have ester bond. Amylopectin cannot have hydrogen bond, and amylose is basically present in starch. It can't have disulfide bond either. We have phospholipids, cellulose, antibody being a glycoprotein can have disulfide bond. In fact, it is stabilized by the disulfide bond. The heavier chains of that antibody molecule. Cellulose is having hydrogen bond. Phospholipids are having ester bonds. Which row shows the features of carbohydrate polymer found inside animal cells. So if you can recall, animal cells having glycogen, it's having alpha glucose, it's having 1,4 branch structure. So you just look for the correct option, and the only correct option that you can find is here. Question 1 till question 10, you can see they are very straightforward question, except for the question of microvilli. No question so far was a tricky question. Now we move to the second 10 questions of the paper. So this is the statement about this. The statement describes the structure of polysaccharide found in cell walls of certain plants. The polysaccharide is composed of 2 different monosaccharides. Is it correct? No, this cannot be correct. It's not made up of 2 monosaccharides. We know certain plant cells. Okay, why 2 monosaccharides? The monosaccharide is joined by 1,4-glycosidic bond. Yes, it's 1,4-glycosidic bond. The polysaccharide contain pentose sugar. No, it's hexose sugar. We know how the plant cell wall is made up of. What the plant cell is made up of. What's true for cellulose? So just recall what's true for cellulose for all of these. We know cellulose is made up of beta glucose. We know it's a hexose sugar. We know it flips. We know it makes hydrogen bond. They are linear structure. There's 1,4 bond only. So if we try to look for the options here, so 1 is incorrect, 3 is incorrect as well. The only option correct is here in 2. So that will be our answer. The general formula of saturated fatty acid given as CH2O and taken oxygen taken twice. So if you look at this formula, it means that carbon and hydrogen are in the ratio of 1 and 2. So whatever the number of carbon will be, hydrogen will be twice of that. So if you look, they are asking which of these fatty acids are unsaturated. So what we'll do, we'll just look for the number of carbons here, and we'll just make up the actual number of what we expect it to be if it's a saturated fatty acid. So if it's this formula, so there are C10H19COOH. How many carbons is it having? It's having 11 carbons. So if it's having 10 carbons here and 1 carbon here, that means 11 carbons. So if it's having 11 carbons, how many hydrogens should it have? It should have 11 multiplied by 2 hydrogens, and that would be 22. And it should have 2 oxygen as well. Is it having 22 hydrogens? No, it's not having. According to our formula of saturated fatty acids, it should have 22 hydrogens, but it's having 19 hydrogen given to us. So this is an unsaturated fatty acid because it's having less hydrogens than we expected. Again, the next is C15, H31, COOH. Now, how many carbons are here? There are 16 carbons here. If there are 16 carbons here, it should have 32, twice of that hydrogens. But again, the hydrogens are 31. Okay, 31 here, and this is the 32nd hydrogen. So there are 30 to 32 hydrogen. Again, similarly, 19 hydrogen over here. There were 20. If you add, there were 20. There would be 20 hydrogens here. So instead of 20 hydrogen for the first, we get 22 here. And here we are having 32 hydrogens, and we are getting 32 hydrogens, 31 and this one. So yes, this is a saturated fatty acid. Coming to the next, there are again 17 carbon and 18 carbon. There are 18 carbons. We expect it to have 36 hydrogens. There are 31 hydrogens here and matter hydrogen here. That makes 32 hydrogens. So 32 hydrogens obviously tells you that this is an unsaturated fatty acid. Coming to the next one, C18, COH, H32. So this is C18 and this one carbon. This makes C19. If this is the hydrogen for this should be twice of 19, which is 38. So is it having 38 hydrogens? No, it is not having 38 hydrogens. It's having 33 hydrogens. So which of these are unsaturated and which of these are saturated? We can just cross one out, and we know that okay, 1, 3, and 4 are unsaturated. Now you know that this question would take about 2 to 3 minutes. So this is exactly how you should be going, because this question needs working. So unless you do the working, you cannot just keep on moving to the next question. So it's best to move on with the question first and come later to the question that need a lot of extensive working, because at the end every question is having 1 mark. So which statement what enable triglyceride to perform their functions in living organism? Triglyceride, we know, just recall this, they are stored fat, they have more carbohydrate bonds for ATP production, they are non-polar, they are non-soluble in water. So I just annotated these points to make sure that I recall what I know about triglycerides. So let's come to the points. The first point is triglycerides have hydrophobic and hydrophilic regions. No, they do not have hydrophilic regions, they are completely non-polar. Phospholipids have both, but triglycerides are non-polar. They do not have hydrophilic regions. Triglycerides have a high ratio of carbon-hydrogen bonds to carbon atom. Yes, that's correct. That's a fact for facts. Hydrolysis of triglyceride release metabolic water. Reason metabolic water, water is added during hydrolysis. Just recall what hydrolysis is. Hydrolysis is about splitting the molecule using water. It doesn't give off water. So metabolic water is not released. The release thing is actually wrong here. So our answer will be true. The diagram show 3 examples of different bond. We already have covered this question in one of the other paper. So you just label the bond, which bond is which. So this dotted bond is a hydrogen bond because it's shown as an electrostatic attraction between hydrogen and oxygen, which will have partial charges. And this is hydrogen bond. This is a peptide bond here between carbon and nitrogen, and this is disulfide bond free. We know that quaternary structure of a protein. Now they have changed the statement. This time they're asking about quaternary structure of protein, which are held together. The quaternary structure is all about R group interaction, or you can say the side chains interact with one another. Now the side chains interaction occur by disulfide bond, by hydrogen bond, peptide bond has got nothing to do with this. So the answer will be 1 and 3. Question 15, which row correctly describe the primary structure or secondary, tertiary, and quaternary structure? We look for what is wrong with each of these statements. Primary structure, we know, is the order of amino acids in which you are joined. Hydrogen bonding actually is about it causes the protein to turn to a secondary structure, coiling. And tertiary structure about R group interaction within a polypeptide, and quaternary is about R group interaction between different polypeptides or a parasitic group. So in A, the first statement for primary is correct. Yes, the term is the folding of polypeptide. Yes, this might be correct because this is actually correct. But the second statement here, that hydrogen bonding between side chains of amino acids is wrong. Hydrogen bonding is there, but not between the side chains. It is between the oxygen and nitrogen, oxygen and hydrogen, which is present in the peptide bonds of the amino acids which are close together. So tertiary structure, and since we know this is wrong, so we can skip this statement. Now come to the second statement. B defines the order of amino acids. Yes, or usually forms immediately after polypeptide synthesis. Yes, that might be that's actually correct because the protein starts to fold into 3D into the secondary coil right away. But 3D folding occurs a bit later. Is held together by all types of bonding that occur in protein. So tertiary structure is held by R group interactions only. So found in globular proteins, we know this is wrong, so we move on. Option C involves covalent bonds only. Yes, peptide bond is a covalent bond, that's a fact, and disulfide is a covalent bond as well. Just recalling that. So it involves interaction between with the hydrogens and carbons. Yes, the hydrogens present in NH2 and COH. Yes, that's correct. Essential for function of enzyme and receptors. Yes, the folding is required. Found by when 2 or more polypeptides join together. That's correct as well. So we know the option C is correct. If we go to option D, involve peptide bond between the side chains of amino acids. Side chains never form peptide bonds. So this is an outright wrong statement. So we'll come to question 16. Now, since colorimeter has been added in the chapter of enzymes, so they gave a question of colorimeter here. How a colorimeter is basically used. So, for example, if you have a test tube here, you place the this is a clear transparent test tube. If you place a light sensitive dye, it changes color from green to colorless. For example, initially the color is green. So this is a test tube I'm having. It's having a green pigment, and this green pigment, if I just irradiate it with light, it turns colorless. So this is what can happen in a reaction. For example, I'm just explaining how the colorimeter might work. So it's actually working. So because that's actually required for the question here, so you should have a basic information. So let's suppose if I have this test tube in which I have a solution which is green in color, and before irradiating any light with it, I'm taking the reading of the colorimeter. I'm trying to note what is the intensity of this color of this green shade, because once it changes the color, I will note down the final reading of the colorimeter, that what's the intensity of the color once light bleached all the green color from here, and how long did it take for the color to turn from this value to this value. Because quantitative values are taken in the colorimeter, you will have the quantified values of this color, how much color is here and how less color is here, how much time you can just what you can use a stopwatch or a timer for how much time is taken for the color to change. So these are some facts that I mentioned here. So what's a colorimeter? It measures the light intensity that can be absorbed by a solution present in a test tube or a beaker, transparent, you can say vessel. A colored solution absorbs more light. This is something of basic physics, I think, and you can just relate to it very well. Clear solution absorbs less light because it will allow more light to just pass through. So dark solution will obviously will be giving a higher value with the colorimeter. Time can be noted for the color change to occur. How can you calculate rate? Rate is always calculated by 1 upon time. You divide 1 by time, and just in case of distance time, so given to you, which they can give in an experiment of photometer or transpiration. So in that you can use the formula of distance upon time to calculate rate. So more of that on later when we come to a question of that sort. So question 16, which relationship could be investigated using colorimeter? The effect of light intensity on the rate at which the solution light sensitive dye changes from green to colorless. Yes, that's very well possible. The effect of temperature on the rate of breakdown of cell membranes and tissue pigmented cells, such as beetroot. Yes, beetroot gives off the red pigment. We all know how beetroot looks like and how it keeps on bleeding its color. The pigment just goes out. So yes, it will be a colored solution. So this can be done as well. 3. The effect of pH on the rate of release of oxygen and the breakdown of hydrogen peroxide by catalyst. There is no color here. This is all colorless. So initially it's colorless. The final thing is colorless as well. What difference will you see? You will see none. There will be no difference in the color intensity or colorimeter reading. The effect of light intensity on the rate of change of skin color of lizard have become paler in bright light. For the colorimeter to work, the light must pass through the specimen. You place the test tube, you allow the light to pass through. Whatever light is absorbed here is given off as a reading, and you just see how much light is allowed to pass through. For example, if it's a lizard, you can't allow the light to cross the lizard and see how much of the light got absorbed in the lizard's body, because there are not just skin cells, there are skin cells, there are muscles, there are bones, there are a lot of tissues inside lizard. So all those will be impeding light. So colorimeter in that skin way cannot be used. So answer will be 1 and 2 only. Which transport mechanism does not require concentration gradient to be present in order to take place? Exocytosis. I think that's something very basic. High concentration of ethanol disrupts cell membrane structure by denaturing proteins and increasing the separation of adjacent phospholipid molecules. As a result, cell membranes can decrease the thickness by 30% and become more permeable. So this is already mentioned to you, that the membrane proteins are being removed because of this alcohol, and between phospholipids they start to move about a lot, and the membrane thickness decrease. Yeast cells can release ethanol as a waste product of metabolism. In response to increased ethanol concentration in their environment, yeast are able to increase the tolerance of their cell membranes to ethanol, which means they must keep their membrane less permeable. They must restrict the movement of phospholipid. They must keep the phospholipids close together. Which statement correctly explains the response of ethanol that could account for the increase in the tolerance of yeast cell membranes to ethanol, decreasing the ratio of saturated fatty acid, which makes the membrane less fluid, to unsaturated fatty acid, which is making the membrane more fluid. So if you have more of these and less of these, so within the membrane, what will it do? It will help prevent the tails of phospholipids on one side of the bilayer from sliding past the tails of phospholipid on the other side of bilayer. So the movement is restricted. This is the major hint that once the movement is being restricted, so the membrane phospholipid will be held together. And yes, that's the answer as well. Let's move on to the others. All others actually are all other options here. They are wrong signs for this observation, because increasing the proportion of a C14 unsaturated fatty acid, which will make membrane more fluid, to a fatty acid which was unsaturated having shorter fatty acid tails, so would increase the fluidity of membrane. We know the shorter fatty acid tails, they make the membrane more permeable. So how can you add a C16 which is unsaturated fatty acid tail to a C18 which is longer tail? So you just need to recall how membrane permeability can be changed by moving to a fatty acid to a fatty acid which is longer, and instead of a shorter one. So in this option, YB is wrong, because they are saying you are increasing C16, which is shorter, because if it's shorter, it will make the membrane more permeable. It would not keep the membrane less permeable. It would not restrict the movement. So this is the wrong option. Activating cell signaling pathways. Cell signaling is not at all linked with the question in any way. Increasing the proportion of ergosterol in the cell membrane prevents the accumulation of polar molecules such as ethanol within the cell. Ergosterol is a steroid, by the way. Ergosterol in yeast, no, you don't need to learn that. Ergosterol in yeast has a similar effect on the membrane permeability as cholesterol in the membrane. So what's the effect of cholesterol? We know cholesterol makes the membrane more fluid. So if it makes the membrane more fluid, so we know that option B and option C, they both are actually telling you the membrane becomes more fluid. It becomes more fluid. So how can the membrane be more tolerant to ethanol? It will become more permeable, and the membrane will actually be promoting the effect of ethanol. So only option A is correct. This question again requires a lot of working, so you can probably just skip that for the moment. If such question comes, then come to it later. I just randomly was drawing this, that you are increasing the for explaining how the ratio of saturated fatty acids is decreasing and the saturated is increasing, and how the kinks in these chains, in these phospholipid tails, if all are saturated, they all can slide past each other very well. But if you have unsaturated fatty acid tails, movement from one layer to the other layer will be restricted, and this is the hint given to us in the option A. That's why option A is the answer. Question 19, in an investigation of plant cell were placed in pure water. You know pure water is having the highest water potential. The initial rate at which water molecules entered the cell R was greater than the initial rate at which water molecules left the cell. We know the cell becomes turgid. What's osmosis? We know solute pulls water molecule, but more solute inside the cell, that would pull more water inside. Water moves into the cell faster initially, and then equilibrium is reached, so it gradually becomes slower. The solution in which it was placed, if the water was entering, the solution must be hypotonic. They're saying in a second investigation, a plant cell of the same type was placed in a solution with water potential equal to that of the cell content, which means it's isotonic. So you know that water movement now will not be in the same rate as it was before. So water molecule will not enter. That's not possible, because osmosis, once the equilibrium is reached, water still entered, but at the same rate. It enters and it leaves at the same rate. So A is not the correct option. Water molecules will enter and leave the cell in equal amounts. Yes, both at the initial rate, that is less than R than the first investigation. Exactly, because in initial investigation R, there were a lot of solute inside the cell that was pulling water, but here the solute concentration is equal on both sides. So the number of solute particles pulling water inside the cell are same as the number of solute particles pulling the water outside. So yeah, the rate would be same. Water molecules will enter and leave the cell in equal amount. This is correct. Both at the initial rate that is greater than R. No, that's not possible, because it's an isotonic condition. Water molecules will enter and leave the cell in equal amounts. Again, this is correct. But both at the initial rate that is equal to R. No, again, this is incorrect. It cannot be greater. It cannot be equal. This must be less, because it is an isotonic state versus a condition in which you are placing the cell in a hypotonic condition. The last question for this video, the protein P53 is produced in the cell response to DNA damage. The protein stops the cell cycle a short time just before DNA is replicated, so the DNA can be repaired. At which phase of cell cycle will this stop occur? So if we know that the DNA is replicated in S phase, we call the cell cycle. We have G1 in the interface. These all are interface occurring, G1, S, and G2. We know that DNA replication occurs in the S phase. You just annotated there. So what will be happening just before S phase? That's G1. So that will be our answer. I hope this was helpful for you. That's it from my side. Thank you so much.